Yusuf, you can start. Now I want to introduce my PhD student, Dr. Yusuf. Many of you know, right? Yesterday you had a demonstration, right? So Yusuf did PhD under me with anaerobic bacterial photosynthetic organisms. That is a Rhodobacter alkali tolerant. So which grows in high salt, which grows in high pH conditions, where you have observed several kind of light arresting complex, change in light arresting complexes, which he is going to discuss. And he did masters and PhD from University of Hyderabad. He is an inspired fellow throughout his career. With this, is a good student. I asked him to present because he is a good speaker. Come.
Good afternoon, and one and all. I'm audible? Fine. Okay. So today, till now, we have you have gone a lot of studies on eukaryotic photosystems like plant and algal photosystems, and so as I worked on photosynthetic bacteria, which is as you all know, photosynthetic bacteria is not only cyanobacteria, but there are some other bacteria also, which is like purple non-sulfur bacteria, anaerobic photosynthetic bacteria, and purple sulfur bacteria are also there. Okay, so I'll take you through one of these bacteria, which is known as Rhodobacter, and it comes under the class alpha proteobacteria, and they are anoxygenic, but they are not, it's not like that they cannot tolerate oxygen, but they like to grow in absence of oxygen, whereas in presence of oxygen they feel kind of stress, and they do not form the photosynthetic apparatus. So title of the light harvesting antenna, photosystem 1, bacterial organisms and algae. So let us see, when we first see the absorption spectrum and the electromagnetic radiation, when we see, is it working, this one? Okay, from here. So when we see at the, no, okay, fine. So let us see at the electromagnetic radiation. So what happens, the energy which comes on the surface on the earth is usually from 400 to it is encompassing UV rays and everything to the far red and radio waves till 1400 nanometer, right? So some part of it which is already absorbed by the eukaryotic photosystems, right? But and eukaryotic photosystems, eukaryotic photosystems are usually able to absorb from above 700, like maybe let's say till 750. After that there is no competition. So what has happened in somehow in nature, these bacteria, where although the energy is very less in these wavelengths, right, because the number of wavelengths, number of waves is lesser, is less in the case of the higher wavelength. Okay, so energy is less, but the quantum or the abundancy is more. So somehow these bacteria have evolved not to compete with the plants, but somehow their ecological niche has also kind of let them evolve, so that they can go to not disturb the plants and other. Let us go for the higher radiation, higher wavelengths. Okay, so in that way you can see there is many species like Rhodopseudomonas rubrum, Blastochloris viridis, Rhodobacter capsulatus, and there are many other organisms and the photosynthetic bacteria which harvest till 1100 nanometer. Okay, you can see here one of the species is Blastochloris viridis, which has bacterial chlorophyll B, bacterial chlorophyll A containing, and there are many other types species of the bacterial chlorophyll A, like A, B, G, E is also there. Okay. So these bacterial chlorophylls when we compare with the plant chlorophyll, you can see the structure of the plant bacterial chlorophyll here. They are relatively more saturated. Okay, the first and second ring of the tetrapyrrole ring is quite saturated. Compared to that, when you find it is not quite, it is relatively more saturated, and so you can so this somehow helps these photosynthetic organisms to absorb the far red region of the electromagnetic radiation. So this is the first component actually of the photosystems. To talk anything, first you should talk about the bacterial chlorophyll. Whereas the tetrapyrrole ring remains same actually, like phytyl chain almost remains the same. So when we see the comparative point of view, if you see what kind of electron transfer, photosynthetic electron transfer types are there in the nature, so you can see the Q type of reaction center and iron sulfur type of the reaction center. Under the Q type of reaction centers comes the oxygenic phototrophs and purple bacteria. So here in purple bacteria, this is the one of the anoxygenic photosynthetic bacteria, which has a very simple electron transport chain. And when you see that this P870 is one of the, it present bacterial chlorophyll A dimer, which is present in the reaction center, from where the charge separation occurs. Okay, so from charge separation it goes to bacterial pheophytin. This is excited state, and then bacterial chlorophyll excited state, bacterial pheophytin, then quinone. And so bacteria doesn't have the plant kind of quinone, it has the ubiquinone, ubiquinone 10 or menaquinone. Okay, so and then from here it goes to QA, QB, QP, and then cytochrome BC1 complex. So bacteria doesn't have cytochrome B6F complex. It is less advanced, less complex actually. It is very simple, and these are the component subunits of the cytochrome BC1 complex. Although some of them are similar to the plant photosystem. Okay, plant cytochrome B6F complex. And then again the electron from here goes back to reaction center by the help of cytochrome cytochrome C2, which is a mobile element. Like, okay, it is it can solubilize, it can like mobile form in the, it can pass from the membrane. It is present, cytochrome BC1 is present in the plasma membrane, right, membrane lipid bilayer. And then from there it takes that electron in the lumen, it remains in the lumen, and from there it again goes to the reaction center. So likewise, oxygenic photosynthesis you already know, so it is containing both Q type of reaction center and iron sulfur kind of reaction centers. So PS1 and PS2 already everyone has talked earlier. And then green sulfur bacteria have the iron sulfur kind of, so only one photosystem is working in this kind of, in these bacteria, whether only green iron sulfur type or the quinone type of the reaction centers. Okay, so moving to next, so I'll be focusing on this kind of quinone type of the reaction center, which is the representative species which is extensively studied is Rhodobacter sphaeroides. So let us see how the photosynthesis occurs in these bacteria. So here is the structure of intracytoplasmic membranes. So in case of the plants it is thylakoids, but here you can see it is the intracytoplasmic membranes. It is a spherical structure and which contains the light harvesting antenna and known as the LH2 and LH1. So these green ones are known as the LH2 and this blue one is called the LH1, and these LH1 actually contain the reaction center. So when you see the structure of purple photosynthetic bacteria, you can see these are the intracytoplasmic membranes which are formed here, and then this is the structure, and this is the LH2. This is kind of LH1, it is present in the dimeric form. So it depends upon sometimes upon the physiology of the bacteria, that physiological condition, that sometimes this dimeric form is present in the form of the monomer or sometimes in the form of the dimer. Okay, so when we see, you can see this one. It is not one. Yeah, sorry. So circularly you can see, and usually it may have 1 or 2 ATP per uh, per ICM, intracytoplasmic membrane, you can say this. So, and when we see the cross section of it, you can very easily see the light harvesting antenna 2 and how the reaction happen. This is LH2, LH2 transfers energy to LH1, and then LH1 transfer energy to reaction center, and from reaction center by the help of the quinone. Quinone here it is ubiquinone. Ubiquinone actually from here it transfers, it like moves within the lipid bilayer and then reaches to cytochrome BC1 complex. Cytochrome C2 is already attached at the luminal side or the cytoplasmic side, you can say, and this is the periplasmic side. Okay. Like here in this case it is periplasmic inverse. Okay, this is remaining towards the luminal cytoplasmic phase. So and then it moves to reaction center. So let us see on the structure of the LH2. Usually LH1 and LH2, LH1 remains bigger in size. Here LH1 is remains bigger in size, and it is made up of the 2 heterodimeric membrane protein complexes known as the LH1 alpha and LH1 beta. Beta is this one, LH1 alpha is this one, and usually the number of the LH1 and LH2 remains around 13 to 14, and some species varies, but not more than 15 or something like that. Okay, and it encompasses the reaction center. Reaction center is made up of the very simple reaction center. 3 reaction center proteins are there, H, M and L, heavy, medium and lightweight. Depending upon their molecular weight, they have been labeled as the H, M and L. And they also have another protein known as the Puffx protein, which actually makes a nick. In the nick means it cuts the circle so that ubiquinone can come and reach, can have the access of the reaction center, and then go to get reduced and then reach to again go to cytochrome BC1 complex. Okay, so this is LH2. LH2's work is nothing but harvest the energy and then transfer to the LH1, and from LH1 it again goes to the reaction center. So LH2 is relatively smaller in size, and it is also made up of the alpha beta heterodimers. This is alpha is the inner protein and beta is the outer protein circle. Okay, and these are very small, 8 to 9 kilodalton proteins, and so this is the transfer section of this is ICM lumen, you can see. So what happens, protons get accumulated inside here, and then this is used by ATP, again proton is accumulated here, and this is used by ATP synthase to produce ATP. So as such in bacteria this is very simple, and its work is only to produce ATP for the expenditure. So when we see the crystal structure of the photosynthetic bacteria, which has been, this is the cryo-EM structure of the photosynthetic bacteria from Rhodobacter sphaerides. We see that dimeric protein reaction center complex, cytoplasmic side, and this is the cytoplasmic view we are seeing from the cytoplasmic side, and you can see 2 dimeric, one dimer is there, and it is containing this Puffx protein. 2 Puffx proteins are there, and how they are interacting. So this Puffx protein has a is nearly 82 amino acid, 8.3 kDa protein, nearly a very small protein, and it interacts with, you can see the interaction, reaction center H, protein, tyrosine, alanine. Alanine is from alpha, and then this is from arginine and glycine, lysine of Puffx. Puffx is red color, so the one which are labeled in red amino acids, they are interacting with the reaction center H and tyrosine and arginine. So these are the interacting partners which make the dimeric form. Okay, so likewise you can also see here the C terminus, like periplasmic side. This was the periplasmic side. So when we just see how this at this periplasmic side, how the interaction is happening here at one side. So So here these are the different amino acids playing important role in the interaction, but these interactions are not covalent, these are hydrophobic, otherwise the salt bridges. So mostly hydrogen bonds are hydrophobic interactions. So then there is another membrane protein which is present actually when we check the different species. So we find that some species do not have the puffex, they have some other protein called the W protein actually. So that W protein is not this much tilted, okay, and it does not make that much bigger nick as we get here, but it makes a smaller nick, but although it doesn't make the dimer. Okay, it is from Rhodopseudomonas palustris. In that, W protein is there, known as the W protein. That W protein tries to make a nick, which helps for the quinone to come and get reduced, but it doesn't help in formation of the dimer formation. Okay, so somehow the physiology of that bacteria is bit different. So that is what I meant to say. When you examine different kind of photosynthetic bacteria, their physiology actually, because of their physiology and genetic makeup, their photosystem structure is different. So let us go puff-Y. Another protein was earlier it was not identified, this puff-Y protein. Why? Because this puff-Y protein was getting masked when they were running the SDS page analysis. This puff-Y protein was co-migrating with alpha protein, alpha and beta protein, because almost there is a very less difference in the molecular weight of alpha beta protein, 8 to 9 kDa, and that protein actually, this also has 5.8 kDa protein. So somehow it was not identified till now, till this paper was published. And so let us see how this protein is looking like. So it actually it has both N and C terminus are oriented towards present towards the cytoplasmic side only, but towards the periplasm it makes a loop, and its interactions you can see here, like these amino acids, how they are involved. Okay, but specifically it doesn't interact with all the alpha beta proteins. It especially interacts with LH113 alpha and LH114 alpha proteins of light harvesting antenna 1. Okay, so there was a lot of study on this, and actually there are many different classes of light harvesting antenna 1 and reaction center, I mean dimeric reaction center classes will get made in the biogenesis of the photosynthetic apparatus. So what happens, many classes like 2 classes were identified in this photosynthetic bacteria. It was their orientation, their point of interaction, like here class 2 and class 1. Class 2 has some tilted orientation, like these two are the LH1 reaction center LH1s, they were interacting somewhat like this. So the angle here formed was nearly 160 or 148, so it was different. So what they proposed, that in the assembly of the reaction center dimeric form, what happens, first the reaction center is there, get assembled, and then after that the puff-X protein which comes and interact with the H protein. We have found the H protein, it interacts with the H protein and then subsequently LH1 proteins get assembled. After that it gets completed, and then comes the puff-Y protein, gets inserted in the structure, and then recruitment of the terminal LH1 subunit. So LH1 subunit gets comes in the very last, and then at the same, then after that when dimer formation occurs, so since there is a lot of interaction happening, right, like hydrophobic or salt bridges interactions are happening. So because of that, what is happening, so it takes some time to reach the final stage. Okay, because of these interactions, because it is a big molecular complex, protein complex is there. So this is an intermediary class which gets formed, and later on structural remodeling, by the structural remodeling it gets into class 1, which is the final class of dimeric form in case of the Rhodobacter spheroides. Okay. So after this, in this study, what they did, after all the crystallographic analysis, they found that what could be the way of quinone, how it comes inside and then how it interacts. So they also did the RMSD simulation, and to find out how they behave in silico, in vitro, how they would be interacting, like how these molecules interact. Okay, so they did, and after that what they found, that during the simulation study they found that Q3 tends to move towards QB. So this is the reaction center, you already know. Reaction center has the two dimeric bacterial chlorophyll, and then after that QA, PA and PB, two dimeric reaction center, dimeric chlorophyll present in the reaction center of bacteria known as PA and PB. Those PA and PB after that goes to QA and QB, these are the quinones. Okay, so quinones and QA and QB. So what happens after RMSD simulation, like they got this structure, but they were not able to see how the quinone, like, so they did simulation tells like suppose if you take a medicine, paracetamol or something, you drug, so there you also people do the simulation, so how the drug efficacy would be there, efficacy in the sense how in your body drug is going to be, like where it will be going, how would be, what would be the fate of it, and how it would be interacting, where it would be going. So by simulation people try to depict that, I mean interpret or like kind of see how it will be behaving. So what they found, that Q3 tends to move towards QB, and QI tends to move towards Q3. Being on their dipole movement is somewhat like that it moves towards the QB and QI moves towards Q3. So in that way, so quinone can come here and interact with the QI, then Q3 and then QB, and then finally move back to go to reaction center and then get reduced and then try to reach to the cytochrome C2, cytochrome BC1 complex, right? So in this way, the reaction and charge separation, after that like electron transfer occurs. So this was the reaction center antenna, reaction center LH1 antenna, and this now LH2. LH2's function is nothing but just to harvest the energy and transfer it to the reaction center by the help of intermediary complex LH1. Okay, so this is the crystal structure of it, and this one is without protein, and this is the cytoplasmic view, and you can see how. So there are two kind of the bacterial chlorophylls. See here there is only one bacterial chlorophyll A, which is able to absorb at 800 nanometer and 850 nanometer. At the same time, it is also absorbing in the carotenoid region. So it is not competing with the plants. Okay, it is taking the far red region, that is 800 nanometer and 8Y, and these are known as the QI region of the spectrum. Whereas this is known as the Soret region. This this is known as the Soret region. So you can see why it could be, because bacterial chlorophyll, how it is present inside, it's about its orientation. Maybe the orientation is the one which is causing this bacterial chlorophyll A, being the same species, able to absorb at the different wavelengths. Okay, so it is the orientation here. Bacterial chlorophyll A 800 is present in this form, whereas bacterial chlorophyll A only 800 is absorbing, is present in this form. This is the carotenoid, which is in parallel to the alpha beta proteins, like alpha beta proteins are standing, carotenoid is also standing here having its interaction, but bacterial chlorophyll A1 is like this, somehow arranged, another one is like this. Okay, so this is what is the arrangement system or nature has made. No, it's not opposite to each other, it's kind of perpendicular, you can say. Only one chlorophyll. I mean, it's one species only, but two copies. So sorry. So further, when we see the pigment protein interaction of B800, so because it also has to get stabilized, it is very compact structure, and it has to maintain its I mean morphology, and because for the electron transfer or the charge transfer, like by resonance energy transfer or something, forced transfer mechanism is there by dipole movement of the chlorophylls and carotenoids. So in that, it has to be maintained. The distance between the molecules has to be is very important, and that will be the important thing to for the molecule, for the charge to move forward and move in a very specific way, very specific speed. Okay. So here you can see this is the interaction of bacterial chlorophyll A, which is absorbing at 800 nanometer, and this is the so it is this is the distance between bacterial chlorophyll 800 B800, and it is interacting with uh, beta protein, arginine and histidine. Okay. So here, and this is asparagine here, this N3, it is so these are the distances they are making. How you can see after the crystal structure, you can see how the distance, how much is the distance, which is like how they are arranged in the photosystem, you can see. And this is bacterial chlorophyll B850, which is interacting with histidine, serine, tyrosine molecule. Okay, so these are the interacting partners coming from the bacterial chlorophyll proteins, alpha beta proteins, and the bacterial chlorophyll A itself. Further, so it is now necessary to know what is the mechanism of transfer. So we have seen that bacterial chlorophyll B800 is like kind of present in a membrane plane, whereas it is parallel, it is perpendicular, the other one B850. So the energy, what happens first, B800, which is more energetic, 800, 850 or 875 or P870. So B800 has the more energy, right? Why? Because it is more shifted towards the left side or high energy wavelength, right? Understood? Okay. So B800, which is having the highest energy, it transfers its energy to B850. Okay, because it has a less energy in comparison to this. And then from here, so what happens, there are so many LH2 and LH1s, and like this will be dispersed, so many LH1 LH2s are there. Okay, and sometimes there will be stoichiometry also, there will be some pattern also. So then energy gets transferred from either it goes to another LH2 subsequent or neighboring one. If it is not there, if suppose some reaction center itself is there, so it will directly transfer here to here. Otherwise, if it is not there, reaction center is not there, it will try to give it to another neighboring one will transfer to the reaction center finally. These are the time 1.2 picosecond is being taken from 800 to 850, and then nearly 5 picosecond from 850 to here B875 nanometer. And then from so here there is a difference, you know, P870 is the charge separation bacterial chlorophyll dimer present in the reaction center. So from B875 it has to go to P870, and it is taking a bit more time, 30 to 50 nearly 30 to 50, 35 to 50 picoseconds it takes to transfer. Why? Because the distance is more here also, and there is a purpose of this for because why it has been done like this, because once the energy comes here, it has to pass here. So once it has passed, and because the distance is more, suppose it has started, it got the energy and it started passing and it got, okay, and it will, the reaction center will start charge separation and subsequent quinone, and the transfer will occur, but that also takes some time. So this distance actually, so if distance is not there, this will keep getting excited and will keep getting transferring the energy to it. So that will get again from energy transfer from here to here is known as the actually trapping. From here to B875 to reaction center is known as the trapping. It is not known as the transfer. It is known as the trapping. Transfer is happening all over here. But here it is transfer from light harvesting antenna B875 to reaction center is known as the trapping. So this trapping, so if it is not far, so detrapping will be more possible. Okay. So in that way, and also it will be possible that they will all getting piled up here if the distance is not less. Okay. So that is somehow the purpose. In that way it has been designed by the nature that the system occurs like this. Okay. And the mechanism which has been proposed for is the Foster transfer mechanism. It is a non-radiative resonance energy transfer through the coupling between the transition dipoles. Transition dipoles is the in the tetrapyrrole ring and also in the carotenoid region. Carotenoids you can found the, because they are the, when there is energy transfer they are in excited states, so dipole movement is there, the main thing, right? So further, so till now what happened, this bacteria can, Rhodobacter can, Sphaeroides can absorb till 875 nanometer only. But there are another species like Rhodobacter blastochloris viridis, not Rhodobacter, blastochloris viridis, which can absorb till 1100 nanometer. Okay, so why is that so? So nature here in this case has given another subunit known as the gamma subunit to the blastochloris viridis, which actually rearranges the system in such a way, means the bacterial chlorophyll in such a way that it is able to absorb till 1100 nanometer as well. It is because of the gamma protein. So you can see the structure and everything is same. Reaction center M, L and H are already there, and alpha beta proteins are there, and out of the alpha beta protein then gamma proteins have also come here. Okay, so in this way what is happening, the rearrangement in the alpha, in this sandwich, actually circular sandwich, the rearrangement is changed, so that is leading to the absorption at more higher wavelength. So further what they did actually, this alpha beta, this gamma protein actually is encoded by 3 operons. Okay, 1, 2, 3 and 4, known as they have labeled is gamma 1, 2, 4. Fourth is actually they have taken a pyralog of it from another bacteria. So what they did, 1, 2, 3 they deleted, and then 4 they have given. So what they are, you can see here, the wild type is absorbing somewhere till here. Yeah, sorry, there it is maximum absorption is till 1018, not 1100. Blastochloris viridis 1018 nanometer. Okay, actually 1100 it goes till here. This is also absorption, but the peak comes at 1018 nanometer, maximum absorption here. But it is not like this, these energies are not getting utilized. This is also getting utilized. Okay, so wild type is having this, but when they mutated gamma 1 to 4, so it has again come back to 950, around 975 here. So when they are again complementing it by the LH1 gamma, so they found it has again shifted. So it is the stoichiometric rearrangement happening within the system, within the proteins, that is leading to the more shift towards the far red region. So here this is the Rhodobacter sphaeroides I grew, and this is anaerobically how I am growing this bacteria. I am not giving a single space to it to absorb, to have oxygen. So if oxygen it gets converted completely in reddish form, and here you can see completely anaerobe is there. So what I say, so there should be one question that like oxygen is also present in the media which I have given already here, right? Because it is also present in the dissolved form. So what do I do with that? Because that may also will cause the trouble for some time, because it will elongate the growth. bacteria won't be able to adapt that easily. So what I did, usually I just inoculate the bacteria and then leave it in dark for some time, nearly 1.5 hours, and let the bacteria just like that sitting and doing the respiration. Okay. So by the respiration it will take all the oxygen dissolved in the media, which while preparing it might have taken all the oxygen, and then I expose to light, then it starts its anaerobic photosynthesis. So then after this I grew the bacteria. This is the TEMI structure. So these are the intracytoplasmic membranes. You can do sonication. After sonication, and I made the gradient 15 to 40% gradient and then loaded on it. So this is special bacterial intracytoplasmic membranes. Okay, I stored, and then after that further experiments can be done. So further, these protein super complexes actually, which we talked, like dimeric reaction center, monomeric reaction centers, they can be separated and visualized by BN page also without denaturing them. BN page blue native is because the color blue, a dye which is used here. So that's why unnative page means there is no denaturation. Okay, as it is, the protein is going to be run, and then in what form, what complexes, because denaturing causes the detachment of the protein. Denaturing means the protein is not going to be in its 3D conformation, right? So here you can see the green gel, completely green complexes. You can see all these complexes, reaction center LH1 dimer, and then ATP synthase is not visible here. You can see ATP synthase has also come, but it is not visible. Why? Because ATP synthase as such doesn't have any pigments. Okay, but after staining I have got this ATP synthase as well. So you can see this is dimer. Dimer has the more molecular weight, so it came up, and then monomers, and then LH2 major and minor are also their species. So that has been separated. So in that way you can do. And further, if you want to, I wanted to go for identification, because here what is this protein which is coming after the after staining. So I wanted to identify, because I didn't have an antibody for this, because bacterial ATP synthase is bit different from the algal cultures. Okay, algal ATP synthase. So and then the 2D I did, and then I gave these protein bands. I just picked it up and gave for identification. So found 4 subunits I gave, and found it to be alpha, beta and gamma subunits having the molecular weight of 55 to 19. Okay. So like this you can do, and then you can also separate these complexes by the help of sucrose density gradient. And so for this, what you have to do, actually you have to solubilize the super complexes. Actually you cannot directly load after isolation. You have to solubilize the complexes, because these complexes are what kind of protein they are? Hydrophobic or hydrophilic? They are hydrophobic proteins. Okay, so they are present in the lipid bilayer. Okay, because they are the hydrophobic protein, they are membranous proteins, so they need a hydrophobic nature environment. So in order to take them out from the thylakoid, from the intracytoplasmic membranes, actually not thylakoids, so intracytoplasmic membranes, I have to provide another detergent, another environment which can mimic, which can give it a house from where it can shift. So you can dissolve it in solubilize it in beta DM or alpha DM or some other hydrophobic detergents, carbohydrates, polymers also. So usually I have used beta DM, endodecyl beta demaltoside, which gives the hydrophobic environment and it dissolves. So then I separated by sucrose density gradient. So here you can see I got the LH2, LH1 reaction center monomer and dimer. Monomer and dimer, you remember, like earlier I told, right? So then I did the absorption spectra, and you can see the absorption spectra here. Like I am getting the 800 nanometer and 850 nanometer bacterial chlorophyll, which we have already seen that the absorption, it absorbs at 800, B800 and B850, and then this carotenoid region, Soret region. And then when you see that dimer has a more density, right? Because it has a more weight. Okay, so it comes in the lower phase, and this lower phase you can just when you can see here the dimeric. So the reaction center phase, it shows that absorption at 875 nanometer. You can see here that it shows that here in these cases dimeric form is present and it is containing the reaction center, because the B875 which absorbs at bacterial chlorophyll B875 is present in this, not in LH2 fractions, because LH2 contains only B800 and B850, right? Whereas LH1 reaction center contains the B875, but LH1 reaction center also contains the bacterial chlorophyll dimeric reaction center, that is in the center where the charge separation occurs, that is B870. So here one can ask the question, why the B870 is not coming? Okay, because B870, it can be masked by the presence of B875, and that too B875 is present in a larger proportion. For B870, only two species of two molecules of bacterial chlorophyll are there, right? Whereas for the LH1 reaction center, what you might have seen in the previous slide, that there are many of the species of the bacterial chlorophyll B875. So when you run the again SDS page, I did this silver staining, and then you can see three proteins only. These are cytochrome BC1 complex protein. Sorry. These are cytochrome BC1 complex protein, and this is reaction center proteins, and probably this is some also cytochrome BC1 complex protein only, but it is in very minor concentration, and here alpha beta protein, and also probably puff-Y may be here, but I am not very sure because the resolution is not very good here. Okay, so like this, so you can also see, like we have already seen, there is a pigment protein interaction, because pigment proteins has to be there, and because it has to be in a compact form, right? So what interaction? So if you have to assess the interaction, how can you assess that interaction? So there is one technique called as circular dichroism, where we use the circularly polarized light. You know polarization, circular polarization, and that you polarization nature of light, that you can tame it according to you, its polarization you can make it according to your wish. Okay, so there the its components, they are traveling in a circular motion, circular way. So in that light, if passes through it, so we can so through that we try to assess the interaction between these pigments and proteins. Okay, so if it is loosened somehow, if it is loosened, so what will happen? The interaction peak intensity will go down. Okay, so here in case of the control condition, like high pH condition where the interaction is too much, like too good, so there the intensity is high, and whereas in normal pH the number is also less. At the same time, interaction may also be, sometimes it is also concentration dependent also. Concentration dependent, not in the sense like you can say interaction only, because here you take at equal chlorophyll, whatever the experiments you do, you take at always do at a same platform. Whatever the experiments, even you have to do in science, you have to give the same platform for everything, whether equal chlorophyll, equal carotenoid or anything. So this is another way. So when we look at the intracytoplasmic membranes, how are they formed actually? So what happens when the bacteria gets divided? Obviously it gets some of the intracytoplasmic membranes by its mother cell, but when the cell elongates its size or something like that, when it matures, it has to make some intracytoplasmic membranes, and which gets formed by the invagination, membrane invagination, plasma membrane invagination, and later on it gets detached from here. So there are some proteins which play important role. Suppose in case of the purple sulfur bacteria, this is purple non-sulfur bacteria which I am talking about, Rhodobacter. Okay, in case of the purple non-sulfur bacteria, there are some proteins known as FMO protein, phenamethylolson protein, which helps in formation of this protrusion. Okay, this invagination, it helps in form. Whereas in this case, in case of the Rhodobacter spheroides or Rhodobacter species, it is the mixed cysti protein which tries to somewhere get here located, and it helps in the formation of the this intracytoplasmic membranes invaginations. Okay. No. Yeah. And when you see this structure, so this is atomic force microscopy images. Atomic force microscopy is another typing method of membranes. By typing method, it will go on the surface of the membrane. Suppose you take out some membrane, plasma membrane, and you want to see the structure or the face of its surface. Suppose this is the membrane I want to see. So the atomic force microscopy will try to give you the surface structure of it, how smooth it is, or is there some protrusion is there, or something is bulging out here and there, or if it is having some shapes here. So that is what they have taken out here. The people have taken out the intracytoplasmic membranes. Sometimes they give mild detergent, otherwise they can go directly ICMs you take, and it will be visible like this. So here you can see these are the LH2s which are present in sometimes pentameric form or hexagonic form, and this is the dimer, how it is just in vivo you can see. Actually just take out the intracytoplasmic membranes and go for the determine some very less concentration, so that it is not very jumbled over there, just very less concentration, so there is no overlapping of the proteins, and then you can visualize it. So these where there is lights coming out na, those are the reaction center. Those are the containing the reaction center, light harvesting antenna one containing the reaction centers also. Okay, so in this way you can see, and even you can visualize the morphology of it and how it is looking like, and sometimes if you give some treatment or some conditions you apply, how the physiology or the morphology will be changing, you can detect it by atomic force microscopy also, and by TEM imaging also. I think I have TEM image. Yeah, I have TEM image also. So this I did the transmission electron microscopy for my cells, and then here also I found this ICMs present here, and also you can see one more phenomena. Actually the bacterial physiology and the shape is also changed, right? You can see something here. So it is because of the high pH grown cells. High pH is actually causing cells not to divide. Okay, so I used the cells gave three conditions, like 3 light intensities, 30, 250 and 500, where in case of the normal pH conditions like neutral, around neutral 6.8 pH, so what happened, the bacterial cells size starts like kind of reduced, and also obviously if the cell size is reduced, the ICM number is also reduced. So what happened here, so later on after the analysis of omics and omics analysis, I found that the cell division assembly is quite fast. I mean cell division is quite faster in this case. Even I was quite confirmed from the from growth curve itself only, from generation time was quite faster in case of the highlight conditions. But I was like after the TEM analysis came, so from the TEM analysis it was quite visible that bacteria is dividing so fast, and highlight is causing actually the bacteria to divide quite fast. And from transcriptome analysis I found that the assembly the septum formation, cell wall formation, like if bacteria has to divide, there has to be some proteins, right? Some proteins, the septum formation proteins are there, they have to assemble. Okay, so like FTSZ protein is there, right? FTSZ protein is the one which is the major protein for the assembly of the septum formation, and so its expression was too high in this case, whereas it was very less, because this high pH retards the assembly of the cell division proteins. So although effect of highlight was there, I found the effect of highlight. Here the cell shape is quite elongated, but it is decreasing with time, with increase of light. With increase of light, the size is decreasing, but its extent is not that much. Okay, so in this way, so if I would have gone, I tried to do atomic force microscopies, but somehow good images I didn't get. So, but one could see for the this also how the physiology of the this how the surface or the face looks like for the intracytoplasmic membranes. So, this is all, and about this rhodobacter, and being very simple, but it has a it can cope a lot of it can grow till pH 10, pH 11. It has its physiology is quite strong and it can cope. So that's all. If anything is there, you let me know.